I think the main concern is the fact that it's a virus that is clinically indistinguishable from foot and mouth disease and all the other swine vesicular diseases. So when pigs present the clinical form of the disease in the field, you can really tell them apart from FMD or the other vesicular diseases such as swine vesicular disease or swine exanthema of swine. So usually animals will present clinical signs will be characterized by lethargy and anorexia, and those usually develop around days 2 or 3 post-infection. After that, we'll start seeing the first signs of lesion development, which usually start with small areas of erythema, either on the snout or coronary bands or the sole of the feet. And those lesions will progress to vesicles, fluid-filled vesicles, typical of the vesicular diseases. Seneca has been associated with neonatal mortality. However, we still don't know for sure if that's actually the case, if it's actual Seneca virus that is causing that neonatal mortality, or if that's just a secondary effect of, let's say, a secondary bacterial infection or a different virus that is there. We know for sure that it is the cause of vesicular disease. The cause of the neonatal mortality that sometimes happens or occurs during outbreaks of Senecavirus is still unknown. We don't know for sure, and that's one of the unknowns is still about the virus. I mean, we did some whole genome sequencing to see if there was anything really striking about the genetics of the virus, and that could be, a factor behind this spike. One of the reasons why a virus emerges is when you see changes in the genome that will increase virulence, for example, and that could be one of the reasons. But although we see differences when we compare the contemporary strains that are circulating since 2014, 2015, we see differences at the genetic level when we compare those to the historical strains. We still don't know if those changes are actually the reason for that increase in virulence. I think the genetics of the virus is pretty similar in the last, from the viruses that originally emerged in 2014-15 here in US until the ones that are causing outbreaks now. I don't think we see a lot of changes in those. There is obviously the normal evolution of any virus, RNA viruses. They're not all identical to each other. So there is some evolution, but they're very similar to the original viruses detected in 2015. In terms of the impact to the industry, I think there are still, people still have concerns, and the virus is still causing outbreaks. There's several reports of outbreaks in slaughterhouses, for example. So after movement of animals to the slaughterhouse, a few days later, the animals that are in the holding areas of the slaughterhouses are breaking. There has always been this association between some sort of stressful situation like transportation or parturition. The original outbreaks were described in sows after farrowing. So there is, a hypothesis that stress induced by transportation or parturition, for example, could be one of the underlying factors that leads to the outbreak. But to date, we don't have any scientific evidence to confirm that or to prove that that's indeed the case. One of the findings on our initial pathogenesis study with the virus was the detection of the virus in the tonsil of all the animals that we inoculated on day 38 or 42 post-inoculation, which is pretty uncommon for any viral agent to remain in the tissue of the host 3 or 4 weeks post clearance of the circulation or resolution of the disease. So there is a question about persistence and a carrier state. Are animals that are infected, can they actually really carry the virus and become persistently infected with the virus and then serve as a source of infection to other animals. So in my opinion, that might be the biggest question that remains to be answered. It would probably change control measures more than our research work, because probably ways of controlling the virus then after an outbreak would be to identify animals that are carriers and removing those animals from the herds. That would be one of the potential examples of what would happen in terms of control of the disease. And this is also a factor that happens in FMD, not in swine, but in cattle. There is evidence that animals that become that become infected, they become carriers of the virus and then could potentially be source of infection. Clinically, one of the differences I think that we see in between the two viruses is the presence of fever in FMD infected animals, which is something that we don't observe in Senecavirus infected animals. The animals will become lethargic, anorexic, and then you will see the development of lesions with Seneca. In FMD, you have that febrile period of 3-4 days post infection. Another thing is the transmissibility. FMD transmits, it's a lot more efficient in transmitting from animal to animal. I don't think we have evidence for that with Seneca. Another important difference is that with FMD, we have several serotypes of the virus circulating, and with Seneca, from what we know so far, and from the limited experimental evidence that we have, we only have one serotype of the virus circulating, at least here in US. We haven't compared anything with Chinese strains or the Brazilian strains. Here in US, apparently we have only one serotype. And that also would have implications in terms of vaccines, should we think about developing a vaccine for Seneca. For FMD, you need to have multiple serotypes within the vaccine so that you provide protection against different serotypes. I think the key is diagnosis. We still have to, if we see a vesicular outbreak or an animal presenting vesicular lesions, we do need to submit samples to the diagnostic lab to confirm that is indeed Seneca and not something else. That will be my number one advice to producers and veterinarians that are working in the field. And the reason for that is because of its similarity to foot and mouth disease. We don't want to waste 1 or 2 days with foot and mouth disease circulating in our herds when we could have detected that a couple of days earlier. So that's one of the main implications of this clinical similarity. We cannot get complacent with a disease like this because of the consequences that the introduction of FMD would have, not only to the swine industry, but to the entire animal health industry in this country.